By simply culturing the total blood with animal serum-free optimized medium in a 75 square centimeter flask, ECFCs adhere to the flask surface. To remove most of the red and white blood cells, wash the flask on day 1 and eliminate the remaining cells by changing the total medium on day 5. Keep screening for colonies every other day or, if convenient, 3 times per week, and you should observe the first colonies around day 12.
Hi, my name is Nicole Hofmann from the lab of Dirk Strunk at the Stem Cell Research Unit of the Medical University Graz. Today we will show you a simplified process for the isolation and expansion of endothelial colony-forming progenitor cells, or ECFC, from adult human peripheral blood. In our lab, we use this process to study the function and biology of ECFC in vitro and in vivo. So let's get started.
This section begins with the preparation and filter sterilization of endothelial growth medium 2, which will be used to culture the ECFCs. Pre-warm 20 milliliters of medium in a 37-degree water bath. Draw 5 milliliters of adult human peripheral blood from the cubital vein into a 6 milliliter preservative-free sodium heparin blood vacutainer tube. Blood samples must be processed within 2 hours of collection. In a laminar flow hood, arrange one 75 square centimeter flask with a vented